This lesson provides an introduction to cell signaling. Here are the objectives. And we're really considering two questions. One, how do cells communicate with each other? And second, how does a signal that's outside of a cell trigger a response that is inside of a cell? Well, broadly speaking, cells communicate using signals, and we can classify those signals into either chemical signals or electrical signals. We're going to deal with electrical signals more when we talk about the nervous system. So for now, in this lesson, we're going to focus on chemical signals. And when we talk about chemical signals, we're talking about ligand-receptor binding. So a ligand is a small molecule that binds to a receptor, which is a protein, and then that triggers some sort of change, delta for change, in the cell that the receptor is attached to. Let me illustrate. Here is a first cell, and it is producing and emitting some sort of signaling chemical. That's this in the red. And then that travels, for example, through the bloodstream to another cell over here that has a receptor protein. This Y-shaped symbol is the receptor protein. And when this molecule binds here to the receptor, it triggers some sort of response within that cell. So this molecule is the ligand. Ligand comes from the same root as the word ligase, like DNA ligase. It means to tie because the ligand is binding to this receptor, and this green Y-shaped molecule here is the receptor. That's a protein that in this example is on the cell membrane, but could also be within the cytoplasm or the nucleus. So ligands and receptors demonstrate specificity similar to enzymes and their substrates, meaning a ligand will have a particular shape and a receptor will have a particular shape, and they will match each other. So ligands and their receptors demonstrate specificity similar to enzymes and substrates. As I mentioned, when the ligand binds to the receptor is going to trigger some sort of action within the cell, and it typically does that by changing shape and or joining up with other receptor molecules and then having further effects after that. So when the ligand binds the receptor, it changes shape or perhaps joins up with other receptor molecules and then triggers further actions in the cell. There are three steps in chemical signaling. There is the initial signal reception. So that is when the ligand binds the receptor. There is transduction. Transduction really means processing, where the signal is taken from one form to another, maybe from one chemical to another, one molecule to another, and then there is the ultimate response. And that response may affect either protein synthesis, for example, a transcription factor may be activated that then has effects on gene expression and protein synthesis, or may affect protein activity. For example, an enzyme may be modified, made more or less active. So the first step is detecting the signal, then processing the signal, and then the last is the response. Let's talk a little bit more about that second step, transduction or processing. Often this has more than one step in it, actually. It's a multi-step transduction pathway. You can imagine one molecule getting activated and then activating several more, and then each of those activating several more. And you can quickly see how this sort of process would allow you to amplify the signal very quickly. So multi-step transduction pathways allow for amplification of the signal. And this is this middle step here. This is happening within the cytoplasm, for example, of a cell that has received a signal. And we'll look in the next lesson in a little bit more detail at some examples of these multi-step transduction pathways. Of note, adding or removing charged phosphate groups to proteins can change their shape by making them more hydrophilic when perhaps they were more hydrophobic before. And remember that proteins, especially enzymes, for example, their function is dependent on their shape, so changing their shape by adding or removing phosphate groups can change their function as well. So signaling cascades, something like this, are these multi-step processing or transduction pathways that allow for amplification in the signal. We'll look at some examples in the next lesson. Now, before we leave this lesson, let's take a look at some categories of signaling chemicals. Now, signaling chemicals can have either local or distant effects. They can have effects by binding to a receptor near to where they are emitted or far away. And in some ways, they're classified based on that. So here are some categories, and you'll see what I mean. Hormones are typically what we call signaling chemicals that have distant effects. Examples would be insulin and epinephrine, and they're produced by specialized cells within structures called glands. For example, insulin is produced in a gland called the pancreas, and epinephrine is produced in the adrenal glands. So hormones are produced in specialized cells within glands in one part of the body, and then they travel in the bloodstream to have effects throughout the body. Now, although the hormone, that signaling chemical, is traveling in the bloodstream all throughout the body, only cells with the matching receptor will be able to pick up that signal and respond. The other cells without the receptor will just be unaffected. It'll be like the hormone isn't there, it's invisible to them. So hormones are one category of signaling chemical where the signal often travels quite far to get to where it has its effect. Another category are neurotransmitters like serotonin or dopamine. These tend to have an effect much closer by. They cross a small gap called a synapse between one neuron and the next and trigger a signal. We'll look at this in more detail in the lessons on neurotransmission, but if this is the end of one neuron, there might be a vesicle here, or there would be a vesicle here with some neurotransmitter in it, and when the signal gets to the end of the neuron for these to be released, and we'll look at how that happens in another lesson, then the neurotransmitters are released into this gap called the synapse or the synaptic cleft, and then they bind to receptors on this neuron after the synapse called the postsynaptic neuron and trigger an effect in that neuron. So neurotransmitters are another class of signaling chemical. Cytokines and calcium ions are the last two categories we're going to look at here. Cytokines, like interleukin, are small proteins that are made by almost all cells, but are especially involved in the immune response. When you have a fever, for example, it's cytokines that are causing some of that response, as well as other processes like growth and differentiation. Calcium ions are predominantly intracellular or within a cell signaling molecules, and they're involved in muscle contraction, which we will look at in more depth in another lesson. And they're actually also involved in the exocytosis of the neurotransmitters that we looked at up here. So when these neurotransmitters were being released into the synaptic cleft, it was actually an influx of calcium ions into this neuron that triggered that. Again, we'll look at that more when we study the nervous system. So that's a quick introduction to cell signaling.